record on computer. All right, let's begin. Good evening. This is Dr. Garais, and this is a video recording of week 2, lecture 2, and of 10 total weeks. This is MED210 Anatomy and Physiology 2. And what you should have done thus far, task 1 was due today. Discussion 1 was due today because it is now week 2. And of course, lesson 1 was due today. For every day, we will be adhering to the university policies regarding late. For every day that it's late, we'll be chopping off a grade right off the top. So please try to get them done by tonight. I'll probably be grading stuff early morning tomorrow or late tonight. So what's this week all about? Now, your task 2, discussion 2, lesson 2, all these submissions, since this is now really a hybrid class, they're all, of course, due next week, June 8th, Monday by 6 p.m. when the class starts. But again, as a public service announcement, I need something. If you didn't attend this evening, I need something until by Saturday, by the 6th. That's when I post attendance. So do a task, do a discussion, and on a lesson. And also, like I said from the first lecture, be proactive with the things that you're doing. So this week we're continuing along the line of immunology. Last week was the first line of defense, which is our skin, the inflammation process, the clotting process. Those are like the no-brainer parts of immunology or the immune system. This week, we're going to be talking about something a little bit more specific. And also this week, I believe the task submission is concept map. If you don't know how to perform a concept map, please give me a call or set up a Zoom session because there are some people who are coming from other universities that already took Anatomy and Physiology 1 at a different university. But if you took A&P 1 with us or any other class, the concept map is the same. So it forces you to group like subjects and like topics together. So the structure and function of the immune system, the different types of white blood cells, and of course the organs associated with immunity. And if you look up here, when you look at what it says read chapter 21 in your textbook, and some people were also asking where the textbook is, when you're on your main page of the MED210, of course, announcements, my Zoom numbers here, my cell phone contact information here as well. But you should be able to find your textbook. Oh, yeah, that's right. Look into your Anatomy and Physiology 1 folder, and there is this thing called OpenStax. So I can show that to you right now. So if I go into my dashboard, which, by the way, you look at my dashboard, it's clean. I have no messages and no things that are due grading. That's the way yours should be. You should always be looking, and look, somebody posted something. You should always be cognizant of what's due and what's required. So if you look at Anatomy and Physiology 1, and you should still have access to your previous Anatomy and Physiology, there's this Anatomy and Physiology OpenStax right here. And you click on that, and or you can go to the openstax.org. Let me click on that. If I go into openstax.org, you just type in Anatomy and Physiology in the search under the subjects, and then you will get the free textbook. But you can use any other textbook. I've had other people in other classes use their Anatomy and Physiology text. There are others who use medical terminology, but the medical terminology is good for the anatomy, but not so good for the physiology portions of the show. So you might have to consider that as well. But if you have a medical assistant text, those of you who are previously from medical assisting or medical billing and coding, that should be more than enough. So let's go back to our class, MED210 for NAX, and let's talk a little bit about what's important. So, oops. Oh, the introduction to the course, I forgot to mention. At the very bottom, under important documents, you click on that. That's where that lab form is. And, you know, the task one, I'm not really going to take points off because that's just the lab form. And I really need that lab form up and running by week 5 because we're already starting to make um, the laboratory schedule. I believe we have a meeting tomorrow regarding the laboratory schedule. So let's jump right in. So task 2, and the first thing we had, of course, you watch the videos and it'll be the same thing. If they're giving you a list of what you need to know, then even if you didn't have a textbook, you can actually just Google it, can't you? So the types of lymphocytes, and that's your white blood cells. So if I look at that, and I could pick out any one of these nice little charts here. Let's oh boy, if you can hear my kid in the background. Where's the chart that I always talk about? Okay, here's like here let's start off with some basics first. Lymphocytes or white blood cells are categorized into two major types: granulocytes and agranulocytes. Now the granulocytes make sense because they got little granules in it. Okay, so those are the neutrophils, eosinophils, and basophils. And monocytes and lymphocytes are the agranulocytes because they don't have any granules in it. So you have your granulocytes here and your agranulocytes here. Macrophage, and now these are not the scale. Macrophages are huge, okay? And that's why they call them macrophage. Macro meaning big, phage, so they're much, much bigger than these neutrophils, eosinophils, and basophils. And then the lymphocytes, we're going to talk a little bit more in detail, or if you could watch the video, watch more in detail regarding B cells and T cells. B cells, they're originally from your bursa, right? During you know, when you're being formed as a young as a fetus, and your T cells are from your thymus, and they both your B cells create antibodies, and your T cells is of course part of your you know, T killer and your T helper system, and we're going to talk about that momentarily. So right off the bat, if you're doing a um, um, concept map, your concept would be, first you have one big circle, meaning types of leukocytes or types of white blood cells. Then it'll be connected to another circle with granulocytes, and then another circle, agranulocytes, and then another connecting into the different types. And since it's also physiology, right? Can you look up all their functions? Okay, and you can make little cards, because you need to know this, because when you do a CBC or a complete blood count, there's something also called your diff, which is your differential. Your differential tells you what kinds of white blood cells are being activated or not being activated. So for example, neutrophils, most abundant type, right? They respond to microbial infection. So if your neutrophils are increased, then you know that it's probably a bacterial infection, and you're going to need an antibiotic. Eosinophils, right? You think parasites and allergic reactions, okay? Basophils, responsibility for initiating the inflammatory response. So if I've got a lot of basophils, I'm thinking what? There's a very large inflammation process in play. Okay, so you could look these up, and also I believe they're also in your notes, but let me go down them anyway. Monocytes, we're talking about largest type. Remember, the macrophages, they're large. Phage, meaning they eat. So their main responsibility is phagocytosis, right? And they have this little analogy here, like the signal man. This is a dendritic cell is what they call an APC, or an antigen presenting cell. So I call it they call it the signal man. I call it the tattletale. The dendritic cell grabs onto something and then it tattles, or it signals the macrophage, which is the big, big cell that eats. And then when it does that, it'll eat the offensive pathogen, or the thing that. And of course, you're gonna get to learn eventually you're gonna get to learn the prevalence on how many of them are normal, but that's in your laboratory class. So you can see here right off the bat, there's, it's better than a textbook because it's already telling you what you need to know. So you need to know what they are, what categories, and what's so special about each one of them. And the video also goes over that as well. But that's a prime example how you could just Google things and use your textbook and then just kind of cherry pick the very important things and the very important parts. So that's types of lymphocytes. Lymphocytic development in bone marrow, and nice to know. But if we look at it here in the bottom, do we really need to know? I need to know the structure and function. And I already got the structure, who are the players in my playbook, and the function, what are the plays. And we now see phagocytosis is one of the plays. Inflammation process is one of the things that we do. Now, lymph nodes, spleen, and tonsils. Now, if you recall your medical terminology, let's look at lymph nodes. Typically, lymph fluid, look at a typical picture, one that's probably on your textbook. Now, if you could see that many of these lymph nodes, they're colored green. Here's a nice picture. Okay, so let's look at this. If they'll allow us. Now, just know that wherever there's an artery vein, there's also a nerve, and also there's also these pathways, or these lymph vessels. And you can see these little beads. Those are your lymph nodes. And they're all around your body. And the function of the lymphatic vessel system is twofold. One, it has to get the lymph fluid to the areas that require increased white blood cells, or fight some pathogens. So let's say, for example, right, you have an infected tooth or something like that. So what's going to happen to some of these cervical chain of lymph nodes? They're going to get full because in order to fight that infection, we need white blood cells, and that's the white blood cells coming from the lymph fluid. And that's also how we also identify cancers, because if you do your breast self-examination, if you could see here the axillary nodes, part of that breast self-examination, if the patient has a non-palpable tumor somewhere there that's potentially cancerous, guess what? The axillary lymph node will be inflamed, and it'll be large enough for you to feel. And that's the reason what the doctor is doing when they're doing the head-to-toe assessment, and they're touching all of these parts. Because let's say, for example, you have a UTI, right, urinary tract infection, some of your inguinal nodes and some of these other nodes here will be activated. Now, it's in green. It's not green in real life. It's, from Anatomy and Physiology 1, when we cut open, for those of us who did have the opportunity to cut open a specimen, right, the pig specimen, you'll see that everything's 18 shades of brown inside. And the spleen and all of these other things are no different. It's just that we use the green color as conventions. Like for arteries are always red, veins are always blue, fat is always yellow, or nerves are always yellow. And that's, it's a convention. It's what artists and who make the diagrams all agreed upon. That's the color it's going to be. Now, the other function of your lymph nodes is also to either carry out excess fluid out of the circulatory system, or put some extra fluid into the circulatory system as well. Now, you can also see the other organs like the thymus, which is the derivative of your T cells. So when you were a fetus, this thymus was very active, and it created your agranulocyte called your T cells. Okay, and there's two types of T cells. We're going to talk about that in a moment. Your spleen. Your spleen is the place where it recycles all the red blood cells, and also it also stockpiles like, I forgot if it's like 20 or 50 cc's, but just a small amount of red blood just in case the body needs it for emergencies. Okay, so you could see why there's splenomegaly in sickle cell anemia, because that's where all the red blood cells have to go through to get filtered out, because red blood cells have a shelf life of about 120 days or so. So they got to go through that. Okay. And also, you can also see that there's lymph tissue within the epiphysis of long bones as well. You can also see there's a ton of lymph nodes in your abdominal area, because that's where all the yucky stuff is. That's where all the bacteria is. But remember, there's no such thing as good bacteria or bad bacteria, just bacteria in the wrong place and in the wrong count. Okay, if anyone recalls my previous lectures. So again, watch the videos. There's a lot more content. And for those of you who find the videos boring, because sometimes they are, let's be honest, remember, watch them in little itty bitty bites, and then make sure you know what you're looking for. So we already talked about the spleen. The tonsils are essentially just enlarged lymph nodes, and they're also part and parcel of the immune system. Now, I wanted to show you where your tonsils are, because a lot of people misidentify where exactly the tonsils are. So let's look at a nice. Okay, see this? This is your uvula. That is not a tonsil, all right? Where are your tonsils? There are these little columns here on the side. So next time you brush your teeth later this evening, take a good look in the mirror, and these are the ones. And also, your mouth is a very, very bacteria-laden place. So we need something that, like your palatine tonsils and your lingual tonsils, and your pharyngeal tonsils all the way in the back there, right? We need those things to safeguard all the bacterial nastiness that are in here. If you've had my lectures before, I always told you that the other hole in your body, your anus, is nowhere near as dirty as your mouth. And we know, because if ever a patient comes into ER with a human bite, or they got into a fight and they scuffed up their hands on somebody's mouth, or sometimes in a fight that people do bite each other, right? Unfortunately, there's a whole bunch of broad spectrum antibiotics and a tetanus shot that the patient has to go through, right? But the only bacteria I'm worried about in the anal region is E. coli, and as long as you don't not wash your hands and make me a sandwich, then that's a problem. So we got your tonsils, you got your spleen, your lymph nodes, your lymph vessels, and I downplayed lymphocytic development in bone marrow. And of course, now you saw a chart with the type of lymphocytes and their major functions. So right off the bat, knowing that and watching this video, hopefully later in your spare time. I love saying spare time, as if anyone has any spare time with everything that's going on. That's what you have to watch. Now, the innate immune system. The innate immune system is what we actually were talking about last week. It's nonspecific. It's what you were born with. So your skin, all these other things, that's part of your innate immune system. But the B cells and T cells, which I'm going to talk about in a minute, that's something very, very different. So right now we have the structure and function immune system. Now we white blood cells and their function, and also the accessory organs. So you could make a pretty decent concept map right off the first 20 minutes of this quick mini lecture. So let's now go to the lesson. Why do I keep on clicking on that? That is annoying. I should click up here, there. Because, you know, MacBooks have a very weird way of scrolling, and I don't have a mouse. So, of course, the other thing that I went through, there's another video, lymphatic system, immunity, and how the immune system works. That's a nice one. This one, definitely know the difference between passive and active immunity. But if you look at this picture right here, passive means you just get it. So what can you get? You just get it either from mommy. That means whoever gave you the immunity, um, uh, it just happened to you. And another way, so you can get it from mommy, which is in utero, or you could get it from actual exposure. You got exposed to it, now you have antibodies, and so now it develops an immunity. Now, well, wait. I think I did it backwards. Let's show this video enlarged.
What's the difference between
Whoa.
passive and active immunity? Well, passive immunity is an example of the body using an army of antibodies.
All right, antibodies, just this pause. Antibodies are these proteins, and they draw them like little Ys because in the shape of the protein, that's what they kind of look like.
Against a pathogen that it has never been exposed to before. So an example is a fetus inside her mother's womb receiving antibodies from her mother through the placenta. Another example is a mother transferring her antibodies to the born child through breast milk.
Now, real quick, that's also how you can transmit pathogens as well. So if mommy's on a substance abuse or mommy's HIV positive, we try not to deliver the baby per vagina, and also refrain from breastfeeding as well. And it's also why we promote breastfeeding in obstetrics, because modern science, we can make everything that's in milk except for the proteins or these antibodies that mommy has. And just as a little side note, it's also the reason why you have a newborn, do not bring them to the mall. Well, nowadays no one brings anyone anywhere, but exposure is a baby, for all intents and purposes, has no real immune system other than the little of it that mommy gave them.
Active immunity, on the other hand, is when somebody gets exposed to antigens, whether it be by an immunization or just entry into the body through openings in the skin.
So what she really means is it goes, so active immunity, somebody purposefully gave it to you, okay? And there's two ways for active immunity. You either get a vaccine, which has an attenuated virus or pathogen. And attenuated means we took out its claws and we only showed the body the antibody. We only showed the body the jacket, so that the body can identify, what the thing is. And they're not easy to make. That's why I was 10 weeks ago, 11 weeks ago, when all these scientists were bragging on how quick they could make a vaccine for COVID, I was there shaking my head. Why did you say that? But again, political pressure and the race for funding created that. So you could see here, passive immunity, relatively short term, active immunity, a little bit longer. And you either have to get a vaccine or you actually got to get sick or actually have to be exposed. For example, and these are little antibodies you see here. And you could see this lovely virus right here. Now that everyone now knows what what coronavirus looks like, all of us protein chemists know that picture, but now the whole world knows it. And now we also know this. These are antibodies. And remember, we talked about the agranulocytes, the lymphocytes, the B cells. The B cells are the ones that get exposed to this pathogen and actually make these. Okay, So now you know the difference between active and passive immunity, and also nice little side note here, where do we find all these antibodies? You find them in your serum, and that's the reason why it's called a titer, or the level at which the antibodies or a substance that's in your body. So we look at the serum, and for those of you who studied phlebotomy, now you know why we have to spin the blood and decant and grab up the serum, because that has all the proteins in it. And one of the proteins that we won't look at are the immunoglobulins, or the immune proteins, and also antibodies. So for example, I spent years in infectious medicine in Southeast Asia with World Health Organization, and I was exposed to TB every day for 2 years. So guess what happened? Did I get TB? I could have, almost, but I was young and I wasn't a susceptible host. And of course, you wear your PPE and all that stuff. And just be precautious, have your universal precautions. But what do I now have since I was exposed so much? I now have a ton of tuberculosis antibodies. So when I take the TB test, you know, that little thing that they put on your arm, my arm turns like bright red and blows up immediately. So no difference between active and passive immunity. That's good. What else? Oh, here. Don't do the lab, but it has some nice notes in it. If you look at this lymphatic system notes, there's some nice notes. Here you go, about the cardiovascular system, parts lymph nodes, some of the stuff that I was talking about. Here you go. Remember I showed you that picture momentarily ago. What is it with me and the English language? Okay, so let's now go look at some of these notes, especially with respect to those agranulocytes. So let's look at, and like I stated earlier, non-specific immune response, or innate immune system, it's not that smart. It's first line of defense, and it's non-specific. But since pathogens are specific, we have a specific immune system, and that specific immune system is called the adaptive immune system. And it responds to antigens through these proteins called antibodies. Now, what's an antigen? An antigen is anything on the outer membrane of, if you recall, all cells have a membrane, a skin. And on that skin, how do we know that the cell is what it is on that skin, there are specific proteins. And those proteins are called antigens. So think of them like, if you've ever watched, like, you know, gang movies, and how do you know that you know, the one gang wearing all red and the other gang wearing all blue? It's because of their coat. And for us, the chemical outer membrane protein coat, and that's called your antigen. And that's kind of how your body knows the good guys from the bad guys, unless you have an autoimmune disease and you have friendly fire all over the place. When you have an autoimmune disease, your body can't tell who are the good guys and bad guys, so your body just attacks everything. But on a normal immune system, your body will recognize these antigens, right, on a pathogenic microorganism, a virus or a bacteria or a fungus. And if they create disease, the body will recognize, hey, there has a specific protein coat, and it will attack it. Now, you have B lymphocytes and T lymphocytes, also known as B cells and T cells. Your B is from bone marrow, also from bursa, but think B, think bone marrow, that's easier way to go. And your T lymphocytes, think thymus, which you've already seen in the lymphatic system picture I showed earlier, right? So what do? Now, I like doing it this way. I like pictures. I'm a pictures guy. And you could also, there's some of these nice, what do you call that? YouTube videos regarding B cell versus T cell. They may be, oh, here's a typical antibody, and you can see it's just a whole bunch of chains of proteins put together, and they come in, and when they're put together, they come into this shape of a Y. Now, where is that? If I could, you know what? Let's watch this video, and then I could break it down for you. Was it this color? Well, we'll see. I think this is the one.
I'm writing my own story now.
Yay, Calvin Klein.
It's okay to be exactly who. So in today's video, we're going to be talking about B cells and T cells. So they're both white blood cells and they have a role in immune function. So that means that they have a protective role in fighting infections. So there are different types of B cells and T cells, but they all originate from the same cell. And this cell is known as the hematopoietic stem cell, which originates in the bone marrow. So through differentiation processes, this hematopoietic stem cell eventually forms B cells and T cells.
Now, if you recall, hematopoiesis, hemat meaning blood, poiesis meaning production of. And stem cells, your body has a ton of them, but the time in your life where your body has millions of them is when you're a fetus. And that's why now the market for fetal experimentation is very lucrative, because if we can figure, like, there are other countries who don't have the same ethical boards as the United States, so it's kind of really unethical to go harvest aborted babies and fetuses and that kind of thing. But you could look at it from a different perspective, that if we can figure out how a hematopoietic stem cell works, or what they call a pluripotent stem cell works, that means if we can figure out the DNA, we can tell the stem cells to be whatever they want to be. So for example, if I don't have enough B cells, do you think I could tell these pluripotent cells, hey, become B cells, hey, become T cells, and then it could cure a lot of a lot of diseases, and it would reverse a lot of these autoimmune problems.
Talk about B cells first. The main function of B cells is the recognition of antigens. After being made in the bone marrow, they travel via the bloodstream to organs like the spleen, or they're located in lymph nodes, and this is usually where they're concentrated in the body. On the surface of B cells, there are these receptors which are called B cell receptors, and these have a function of binding onto antigens. If you don't know what an antigen is, imagine it's like a small chemical or molecule which is on the surface of harmful bacteria or pathogens, or it doesn't have to be on the surface of a pathogen, but the body recognizes it as a foreign substance. Once an antigen is bound onto the B cell receptors, the antigen is engulfed into the B cell in a process called receptor-mediated endocytosis. The B cell then breaks down the antigen, and the breakdown products are then placed on the surface of the B cell in a special type of molecule called a major histocompatibility complex. And in B cells, it's the second class of major histocompatibility complex which is present. The reason that this occurs is it allows helper T cells, which are also known as CD4 lymphocytes, to recognize and bind onto the major histocompatibility complex class 2. And once it's bound, it releases lymphokines. Lymphokines are important because it causes this B cell with these specific B cell receptors to clone itself via the process of mitosis, so that we have more of these B cells with the same receptors. And it causes the B cells to differentiate into plasma cells. And the main function of plasma cells are to release antibodies.
Okay, let's stop. That's a lot of stuff. But it's a lot of stuff that's important, because when you look at immunologic assays or immunologic tests, there's these things that they all measure. So they'll measure B cells. Another thing they'll be measuring is MHC or MHC2, right? Major histocompatibility complex. This is all. MHC also deals with why, even if you have a twin, you can't just give them your kidney, because it'll reject it, because you could see how highly specific your B cells are. And the MHC2 complex, right? Along with the T helper cells. Now remember, the T helper cells is part of the T cells, but they help. And also another thing we can also measure in the bodies are lymphokines. Lymphokines, of course, tell the B cell to go make plasma cells, and these plasma cells make antibodies. And then the antibodies will attack this antigen without all this process. So that's why they call B cells memory cells, because they remember. They don't have to go through all of this process. They go through all of this process the first time when it's the first time they get exposed to this antigen. That's what's really cool about B cells. Now, T cells, you may have heard T cells or CD4 cells or T helper cells. T cells or helper T cells or CD4 cells, those are get really low in HIV. So now it makes sense. If I have HIV or my patient has HIV, they don't have any T helper cells. Without any T helper cells, can any of this process move forward? Nope. That means if you have a pathogenic antigen, there's no reaction, there's no, and you don't have any specific line of defense to create antibodies. And then the patient can get very, very sick, and that's full-blown AIDS. All right. So that's the connection on why B cells are important, and also the CD4 plus cells, also known as your T helper cells.
Antibodies that are produced by the plasma cells are specific to the antigen that was initially bound onto the B cell in the first place. Now let's talk about T cells. We just mentioned one type of T cell called the helper T cell, also known as CD4 T cells, and just to recap, it binds onto the second class of major histocompatibility complex, which is a molecule located on the surface of B cells, and this binding causes the release of lymphokines. Other types of T cells include the cytotoxic T cell, also known as CD8 T cells. In the case,
these cytotoxic T cells, they're also called T killer.
Cytotoxic T cells, or CD8, imagine you have a cell infected by a virus. The cell is now infected and it contains viral DNA and viral mRNA. Because of this, the virus is now making viral proteins and peptides. Since all cells with a nucleus have major histocompatibility complex class 1 on their membrane, these viral proteins that the infected cell is making is presented by the class 1 major histocompatibility complex molecule on the surface of the infected cell. So even though that sounds very confusing, imagine the infected cell is kind of giving a warning signal by showing the infected proteins on its membrane. The cytotoxic CD8 cells have T cell receptors on their surface that recognize the peptide on the surface of the infected cell is not normal. So it binds onto it, and when it binds, the T cell either secretes cytotoxins like perforin or granulysin, which initiates programmed cell death in a process called apoptosis. Or once the binding occurs, the T cell expresses a protein called Fas ligand or FAS ligand, which binds onto the FAS molecule present on the infected cell. And this forms the death-inducing signaling complex. And eventually we have apoptosis of the cell. We also have...
All right. So what is apoptosis? Cells have a certain life. Like I mentioned earlier, red blood cells have a shelf life of 120 days. So typically, a cell would keep on living longer. But the cool thing about cell-mediated immunity, or immunity via T cells, right? The infected, the CD8, the T killers recognize the bad, the virus being produced in an infected cell. So you could see how it also acts like the T killer does two things. One, it'll tell the cell, hey, it's time to die, right? Via these things, cytotoxins and FAS ligand. That's just fancy. They're fancy chemical stuff. Just know that the CD8 will attach onto this, infected cell, and it'll tell the cell, and that's called apoptosis, hey, go die. That's one. Now, the other thing that the cytotoxic T cell will do is along with the T helper, it will also activate the memory cell, because then they were like, hey, remember this guy from before? And then the antibodies will go and then will consume and eat this infected cell. So you could see how, even though we talk about the B cells and T cells as two different things, B cells is called humoral immunity and your T cells is called cell-mediated immunity, they're kind of linked together. And you could also see the other markers that we could also look at, not only for antibodies, we can look at MHC class 2, MHC class 1, cytokines, cytotoxins. So these are all related to the tests that we look at to make sure that my patient is not immunocompromised. And if none of these things are present, they'll show up in laboratory tests. And all of these proteins happen to be in the serum of your blood. All right. So that's like a nice video of all these words. It's nice having the words, right? And this is antibody hooked up to antigen, but you saw that as a little bit better picture. Why does it do that? So that's T cell mediated immunity versus B cells. Now, real quick regarding ELISA, because everyone's talking about viruses and things like that. How do you detect the presence of a specific antigen for something? You attach an antibody onto something. First of all, what does ELISA stand for? Oh, where is it? Acronym for ELISA. Enzyme linked immunosorbent assay. Okay, so what does that mean? We use enzymes and then we link it to antibodies. So let's say, for example, let's look at this antibody. Let's say, for example, like this is a cancer cell that has bound antigen. And then I take these antibodies and then I attach something onto it, like radiation. I did my graduate, my postgraduate, pre-doctoral and some of my postdoctoral work in protein chemistry. And one of the things that we look at is that we want to see how antibodies like these stick onto antigens. And one way to identify the bad guys is hooking something that will either the star, like it'll change a secondary antibody that will either change color, or it has an enzyme that will also maybe release some radiation, and we could count how many bound versus unbound antibodies. So ELISA is used a lot in experimentation, and it's also used a lot in diagnosis of many diseases through identifying their specific antigen. Now, what else can we talk about? Nope, now it goes into the lab, and we don't want to talk about that. Okay, where is, great, I closed all the screens. So in conclusion, last week was non-specific immunity, which is first line of defense. And this week was specific immunity, which is last line of defense. And you could see how diseases like autoimmune disease and HIV are very, very deadly to the system, because it interferes with the normal processes. And isn't that what pathology really is? It's just the interference of normal. Okay, so lymphatic system, we went through that, we went through this, and what's due for next week? Of course, your task, which is the concept map discussion, same protocols as always, 200 words with APA cited APA citations with proper format. Now, Of course, we all now recognize this. And now you know exactly what these are. These are specific proteins that are on the surface of this particular pathogen. And this pathogen is HIV, and is also part of the Coronaviridae family. And this is what, in theory, antibody or an antibody can latch onto, because these things present as a coat or an antigen. And remember, the gang colors analogy. So, wait a minute. Let's go back. So, of course, we're in lesson 2. It's here in the application assignment. So you don't need to do the lab. Every week is application assignments for lessons. And you click on this, download the case, and then, of course, read the case, and then answer the questions. Answer the question for part. And also, it has some really nice pictures now that you have a primer on how the T cells and B cells work. Now the questions should be relatively easy. And questions 1, 2, 3, 4. I think there's only 4 questions. Yep. So answer these 4 questions. They're due next week. And please put it in a Microsoft Word document and make sure the document has your name on it and the file also has your name on it. So because when I open it, I sometimes save them for future comments or whatever. But yeah, it's easier to identify. Now, our class is relatively small, but you can imagine if you have a class of 25, 30 people, if you don't properly label your file, your professor might use it. Okay, and now just real, no, I don't want to talk about that. That's too much. And you can see how the glycoprotein coat, GP120, which is 120, 120 like monomers long, or little Lego pieces long. You could see how they made it like a key. And HIV is a reverse transcriptase, meaning you see that word ACE means an enzyme. It can take the normal DNA to RNA sequence, and they can make it backwards. So it can take your RNA to tell the DNA, hey, erase all the plans. You're now no longer going to make any proteins for your cell. You're going to now just make HIV virus. And also, let's say, for example, the virus is inside the nucleus of the cell. And now you know how immunology works. It has to work for recognition. If something's inside the cell, it's like a home invasion. You don't know that there's something going on, and the cops or the macrophages have no way of knowing that there's something bad until when the cell explodes and then releases a whole bunch of virus. So that's why viruses are very, very difficult to cure. They're very difficult to handle. And the world now already knows that by practice. There used to always be a joke about doctors like, oh, why can't you guys cure the common cold? Guess what the common cold is made out of? It's made out of a particular strain of Coronaviridae, part and parcel of the COVID family as well. So you can only imagine if we don't, because we don't even have 110% reliable vaccine for the flu, or have no real cure for the flu, which, by the way, last year killed more than 27,000 people, right? But no one bat an eyelash about that, right? And remember, everybody, please, whatever's going on in the world, please be scientists. Please look at data and make your decisions based on data and based on facts, based on .org, .edu, right? Just like the way we find the proper APA format for our discussions. All right, now I can unmute you guys if anybody has any questions. There's only 2 of you. Miss Sabrina, Miss Sonia, do you have any questions? If not,
No.
oh, if not, you know what's due next week. And if you need any help or tutoring, quick Zoom sessions, I've been promoting that. I did like 10 of them last week. and it's only like 15-20 minutes, and it solved a lot of really quick problems. Or if there's a video you want me to make on a specific topic, I can easily do that. All right, ladies, you automatically have attendance, but again, thank you very much for attending, and I'll see you guys next week.
Thank
Okay.
you.
Thank you, Dr. Rice.
Have a good night.
Good night.
Okay.